Deep within your cells, a silent executioner waits in the dark, poised to dismantle the internal machinery the moment it receives the signal to strike. These lethal operations occur on a massive, invisible scale, millions of times per second. If this internal recycling protocol fails to clear the way, the cell quickly burns through its own resources and starves. This is no random biological chaos. It is a perfectly targeted assassination. But how does this hitman isolate its specific victim? The groundbreaking study from the Max Planck Institute, led by Professor Brenda Schulman, has finally tracked the hitman to its source. Capturing this microscopic strike required specialized high-resolution imaging technology. For the first time, we have the complete 3D atomic blueprints of the entire process in action. The trigger is a state of emergency. When a cell runs out of vital nutrients, it enters a phase of metabolic starvation. This crisis activates LRRC58. Its intensity spikes the moment the cell begins to struggle. The objective is clear: hunt down and eliminate the internal components that are needlessly draining the cell's final reserves. The proof of the kill is undeniable. As LRRC58 rises to its peak, its specific target is entirely wiped out. The target is CDO1. The hitman does not grab it randomly. The two must align like a perfect lock and key. Our 3D models reveal the exact structural geometry of this lethal embrace. The hitman forces CDO1 into a specific orientation, exposing one single structural weak point, lysine 8. Hide that one microscopic spot, and the natural hitman fails to strike. By capturing these 3D maps of nature's own protein-destroying machinery, we discovered the secrets needed to hijack the system. We are no longer just observing a cell's natural internal recycling process. We are learning how to actively program it. We can now apply our own labels to force any protein into the cellular shredder. This is the role of molecular glues. They bypass the strict requirements of nature, forcing a connection where none existed before. This synthetic glue creates an entirely new, unnatural point of contact between the hitman and its target. The result is absolute. The cell shreds the target indiscriminately, ignoring the natural safety locks that protected it only moments before. These atomic maps have moved beyond simple observations of the natural world. They provide the exact blueprints to rewrite cellular behavior, allowing us to build drugs that command a cell to destroy its own disease. Read the full paper to explore the atomic structures of the LRRC58 CDO1 pathway and the future of targeted protein degradation.